All right, welcome everybody. We're going to do some bioinformatics today. In particular, this is for the Amino Labs PCR-it Kit, and our goal today is to use a program called ApE, or APE, stands for a plasmid editor, to model a PCR reaction so that we can calculate the DNA fragment sizes that we'll hopefully get in the PCR reactions that you're going to run in the classroom. Okay, so I've already opened up APE. There are instructions on how to download it. You'll need a normal PC computer or a Mac computer in order to run the software. So yours might look a little different than this if you're on a PC, but overall the different tools available should be the same. I've got a folder here with some files in them that are available for you to download from the Amino Labs website. And these folders contain DNA sequences relating to the DNA templates that are included in the PCR-it Kit, as well as the primers that are in the PCR-it Kit. We're going to go through one of these two today in this video, and the other one you're going to have to do on your own. All right, so I'm going to open up a few sequences here by double-clicking. All right, and I'm going to separate those out, and I'll describe what these are. On the left here, we've got the DNA sequence for a plasmid, a DNA plasmid, called PBR322. Okay, so what you're seeing on the screen are the A's, T's, C's, and G's, the nucleotides that make up the DNA strand within this plasmid. Okay, keep in mind that this DNA template is double-stranded DNA, so it's going to look like a beautiful double helix. And one more thing to note here is within the sequence, you'll see some different colors. So you'll see the yellow, the green, the blue, and these are just annotations that scientists have added to the sequences to know what that region of DNA does. So you'll see it here, you'll see it in the plasmid, and that's all that really means. All right. I'm going to click on this little plasmid symbol here and just give you a quick look. So it's a circular piece of DNA. Within the tubes in your PCR-it kit, there are going to be thousands and thousands of these little plasmids floating around. And this is what you're going to amplify DNA from. Okay, this is the template. Now we're not going to go through it today. That's for another discussion. Today we're doing the modeling, so I'm going to close this and let's look at these other sequences. So you'll see one is called PBR322FWD, which stands for forward. That indicates that this is a forward primer. And we've got PBR322REV, which stands for reverse. Okay, this is the reverse primer. And together, the reverse and forward primer will bind to the DNA template and allow you to, or enable you to, amplify the region in between these two. Okay, and now let's go ahead and model what the size is going to be of the DNA fragments within your PCR reaction. So I'm going to click on the tools bar on the top. I'm going to click PCR reaction. It's going to open up a dialog box here. And at the bottom, we've got a bit of a visualization of the DNA template. Rather than being circular, it's showing it here as linear, which is fine. When we click on template, we see a few different sequences here, right? We want to be on the .gb, which is the full sequence of the template. Okay, we don't really want to be clicking here on these primers. If we go to the database here, we can add primers from a sequence. So let's go ahead and add those. You'll see them show up here. I now have the forward primer and I've got the reverse primer. And again, I've got the template set to be the full sequence of the PBR322 plasmid. Okay. I'm going to click on this button here for select primer pairs, and you can see right here it says there's no forward primer and no reverse primer, and so I'm actually just going to click on the forward, I'm going to click on the reverse, and they'll automatically populate down here. And if we look into this little box here, we can actually see these little arrows here. They show where the forward and the reverse are. So within the entirety of this template, we can actually now visualize where amplification would happen. So within the PCR reaction, it's going to amplify, the polymerase is going to amplify this region in between these two arrows, okay? But let's have a look to see what the size of that fragment or those fragments are going to be, because it's going to make many, many, many of those fragments. So let's click on New PCR Product. And it opened up in a different monitor here. Let me drag this up here. It'll be a little bit on the behind until I close this dialog. And let's move this to the front. And there we are. So now we've got the actual DNA sequence that will be amplified from this DNA template. Okay, we can see here that the sequence, the theoretical sequence, is going to be 773 base pairs long. Okay, and so this is pretty important. This is going to be used within your analysis when you run your agarose gel to confirm that you successfully did a PCR reaction and got the correct size band. And we could take this one step further and model this in an agarose gel. Let's click on the agarose gel up here. I'm going to click on this on the checkbox here so it goes on to lane 2. And I'm going to click OK, and it's opened up on another monitor again. Let me drag that up. And this is what it should look like in a gel that has a 1 kb plus ladder. Now, keep in mind, not all ladders are the same. So this may not be exactly what it looks like, but here it shows, it designates what these different bands are. So this is the 100 base pair band, 200, 300, 400. Here's the 650 band, and here's the 850 band within this custom ladder here. And so you can see that your band, which is 773, should run in between the 650 and 850, okay? Again, the ladder you're going to use within your agarose gel experiment will be different than this, and you'll have to see in the paperwork that comes with the ladder what bands are what size, okay? And then from there, you'll need to find out, is your 773 base pair band running in between, let's say, for example, 1000 and 750, right? So yeah, there you go. So we've gone through the modeling of the size of your PBR322 PCR reaction. You can go ahead and save this file and keep it and put it in your notebook. You can print it out. I'm going to close it. And also you can save this file of your PCR product, name it properly, and you can either print that out or just hang on to that as evidence that you've done your modeling. All right, so with that, we can stop and you can move on to doing the modeling of your other PCR reaction. So this would be the PUC19 reaction. And with those two, you'll know what the two fragment sizes should be within your PCR-it Kit reactions.